We're going to learn how to solve Mendelian monohybrid cross so that we will know how traits are passed from one generation to another. It's Mendelian because we're going to be using the idea, the concept of Mendel fused with the current understanding of how traits are passed, how genes are passed from one generation to another to trace how cystic fibrosis can be inherited. It's monohybrid since we're only going to be using one trait, and this trait is called cystic fibrosis. This is a disease of the respiratory system caused by a mutation of a gene. The name of the gene is cystic fibrosis transmembrane conductance regulator gene, or CFTR gene for short. Once a mutation of the gene occurs, it disrupts the normal production or functioning of the CFTR protein in the lungs as well as other parts of the body. So the unmutated form codes for a normally shaped tube. This is the phenotype, and the gene carrying the CFTR is the genotype of the individual. So this tube is mainly made of protein. So this is the tube. It's hollow, it's cylindrical, and then it allows the substance called chloride ion to move in and out of the cell. And that allows the mucus to be sticky and easily coughed out, which we call phlegm. The mutated form of the CFTR shows a phenotype of an abnormally shaped tube. So this is the unmutated form, the expression of the unmutated form, and this is the phenotypic expression of the mutated CFTR. So look at the size of the chloride ion and look at the inner lining of the tube. Once a mutation occurs, the chloride ion cannot easily pass through because of its size and because of how the linings of the tube is structured. Once this happens, mucus becomes crusty and waterless, it's dry, and it's difficult for that person to cough it out. That's one of the symptoms of cystic fibrosis. Now, cystic fibrosis, or this gene actually, is found in the chromosome number 7, and it's somewhere here in the lower form or lower part of the chromatid. Now, cystic fibrosis causes a lot of mucus to accumulate in the bronchioles of the lungs. Look at this. And that's why the person has difficulty in breathing in and breathing out, difficulty in breathing. It's because of the mucus that blocks the passageway. This is a normal pathway where gas can flow through easily and you can breathe without pain. Persons with cystic fibrosis is always in a lot of pain because mucus accumulates and this needs immediate attention or medication. Now, of course, once this happens, blood is oozes out from the capillaries because there's pressure of the mucus, and it is prone to bacterial infection. And people, if the person is left untreated, can die. Okay, and the cause of this suffering is just one small part, one small structure of the body called the CFTR gene. Now, this cystic fibrosis is a recessive disease. It means that a person must have a mutation must inherit two copies of the mutated gene to suffer from CF, meaning um, the person must receive one allele, mutated allele from the father, and one mutated allele from the mother for him to show manifest cystic fibrosis. However, if that person receives only one mutated allele, let's say from the father, and one unmutated allele from the father, remember in the law of dominance that um, the dominant covers the recessive. So the person who has received a mutated recessive gene cannot show this because there is a dominant trait. The person is healthy. So that's the law of dominance as discussed by Mendel. But a person who has one mutated allele but doesn't show cystic fibrosis is known to be carrier. So let's just have one sample problem. If the mother has no mutated gene for cystic fibrosis, capital C, no mutated gene, so that person is homozygous without cystic fibrosis, while the father is a carrier of cystic fibrosis. Carrier means directly heterozygous without cystic fibrosis. What is the chance of the offspring that will be normal and healthy, like phenotypically normal and healthy? The person is a carrier, that offspring, or an offspring having cystic fibrosis. Now, in our case, the symbol for that gene is capital C for healthy or no cystic fibrosis, and small c for having cystic fibrosis. However, if some problems that you've met along the way does not give you this, then you have to follow the rule. In our case here, it's given, so we also have to follow this one. Capital C for normal healthy offspring and small c with cystic fibrosis. Next, we're going to identify, for step 2, the genotype of the parents. And commonly, it's given in the problem. So here are the genotypes of the parents: homozygous without cystic fibrosis, so homozygous without cystic fibrosis. So the genotypic symbol is capital C and capital C, while the father is said to be here heterozygous without cystic fibrosis. Again, take note, the father is heterozygous, he's otherwise known as carrier. So without cystic fibrosis, so it's capital capital C and small c. Capital and small, hetero meaning different, heterozygous. So step 2. Now for step 3, we have to segregate and assort the alleles, and they can be carried by or found inside the gene, no, the parent sex cell. So just to mimic how segregation is done, all you have to do is just separate capital C, which means this C gene or allele will be part of one egg, and the other one will separate and it's also going to be part of the other egg. So each allele segregates and found in the eggs, no? To each of the eggs, sorry. And just to show that they have segregated, you just can use and or comma. So in the father side, so capital C is part of the gene in the sperm cell, and the other small c will be found as part of the allele component of the sperm cell. Okay, so what we see is the law of segregation, in which genes or alleles segregate from each other. Now, step 4 and 5 is uh, we'll be using the Punnett square. So a Punnett square is named after Reginald Punnet, and it shows the genotypes, possible genotypes of the offspring of the which comes from the combination of the parents' gametes. Step 4, all you have to do is place the gametes carrying the symbol in the row heading for the female and the column heading for the male. Well, step 5 is simply combining the alleles of the parents. Okay, so this is step 4, and here, okay, step 4. Now, step 5, all you have to do is to combine the gametes. These are the chances that this egg might be fertilized by this sperm carrying the allele coding for CFTR gene. So here's the first possible combination, and this one. And once fertilization happens, meaning a new offspring, a new organism is formed, that organism will be carrying this genotype. Okay. There is also another possibility that this egg will be fertilized by the sperm. And the genotype of that offspring formed is capital C, the gene from the mother, and the gene from the father, and the genotype of the offspring. So just take note that they are both capital, capital C, capital C. And then there is also a chance that this egg will be fertilized by this sperm carrying that specific allele. Okay, and a new organism is formed, and that's the genotype of the organism. And of course, there's a chance that this would be fertilized, this egg will be fertilized by this sperm, and a new organism is formed, and the genotype of the offspring will be capital C and small c. Okay, so step 4 and step 5. Once it's done, you go to step 6. Step 6 is calculating the percentages of the phenotype and genotype and indicating the ratios. But we have to interpret what's in the Punnett square. So let's start with this capital C and capital C. Remember, both capital letters, so we'll have it homozygous. Homozygous. And since capital C represents without cystic fibrosis, so we add without cystic fibrosis, and the genotype of this genotypic symbol is homozygous without cystic fibrosis. Capital C and capital C. And I'll mark here as 1. And then here is a same genotype, and we'll mark it with 2. Okay. Now the next genotype is a different one. So we take this in a different way. Capital C and small c. So capital and small hetero. And remember, all heteros are carrier for cystic fibrosis. But which one? This is capital C. It represents without cystic fibrosis. And this one is small c with cystic fibrosis. But remember in the law of dominance, that the dominant trait covers the recessive. So what we write here is heterozygous without cystic fibrosis, 1. And of course, here is another one, so we'll have 2. Okay. So how do we get the percentage? Remember, percent is 100. So we'll have 100 divided by 1, 2, 3, 4 boxes. So 100 divided by 4 is equal to 25. So each box is 25%. 25 plus 25 plus 25 plus 25 is 100%. So if we take homozygous without cystic fibrosis, 25 plus 25 is 50%. And here, it's heterozygous without cystic fibrosis, 25% plus 25% is 50%. So the ratio of the genotype is 2 is to 2. Okay, we got 2 from here and 2 from here. So we're not using the lowest form of the ratio. Okay. Now, next we identify the phenotypes of the offspring. So we can use this data or this data for the phenotype. Now, here, what would the phenotype be of the offspring? So without cystic fibrosis, so this individual will exhibit no cystic fibrosis. This one, capital C and small c, without cystic fibrosis, it's a dominant trait, so we'll have no cystic fibrosis, that would be 2. And this one, of course, no cystic fibrosis, that would be 3. And again, this one, no cystic fibrosis, 4. So in fact, we don't have a ratio, um, 4. But if we take a look at the percentages, it's no cystic fibrosis is 100%. So this is step 6. This is the result of our genotype. So step 7 is to answer the problem question. So what would be the chance of the couple to have phenotypically normal or healthy offspring? So offspring phenotypically with no cystic fibrosis is 100%. So all their children, whether male or female, will all be healthy without cystic fibrosis because their gene contributed shows the dominant form. Now, what is the chance for a carrier? So we're talking about here the genotype. So a carrier is heterozygous. So the carrier is 50%. So to interpret this, it means that uh, if that couple will have either male or female offspring, that offspring has a 50% chance. There's a 50% chance that he or she is a carrier. She might be carrying the allele which expresses cystic fibrosis. She might. So that's why it's 50% or he might. However, there is also a chance that that offspring will not carry the allele because it's a 50-50% chance. Now, how about children having cystic fibrosis? Having. So it's showing cystic fibrosis. It's a phenotype. Therefore, there is no chance. So this couple can breathe free because their children will be healthy, but they will know that that gene of mutated cystic fibrosis has a 50% chance of being received by their grandchildren. So that's how we trace how traits are passed from one generation to another using the monohybrid cross and using Mendelian pattern of inheritance. However, not all, take note, not all of the diseases or traits are passed this way. So this is just one simple trait following the Mendelian laws of inheritance. Thank you.